Well, this lecture is the second installment of the material on the gastrointestinal system, and we will be discussing primarily the processes of digestion and absorption. The GI system digests the three basic biomolecules: carbohydrates, that is sugars and starches, proteins, and lipids, which of course are what we generally refer to in diet as fats. And of course, the breakdown products of this digestion will be absorbed, as will vitamins, minerals, and water. So we'll look at each of these three biomolecules and how they are digested next. First of all, for carbohydrates, the typical diet consists of about 250 to 800 grams of carbohydrates. Most of the carbohydrates are consumed as disaccharides or polysaccharides. When we refer to a saccharide, we're referring to a sugar molecule. So a disaccharide is two sugars, and a polysaccharide, of course, is more than two. The disaccharides that are primarily in our diet are sucrose, lactose, and maltose. And the polysaccharides, which are primarily storage forms of carbohydrates, are starch, which we derive from plants, and glycogen, which we derive from animal tissues, such as meat. There's also cellulose, but which this cannot be digested. This is what we refer to as fiber in the diet. So, of course, many fruits and vegetables contain a fair amount of cellulose. If you want to try to think about what cellulose really is, think of something like a stalk of celery, which has a great many of very robust fibers in it, and that gives you an idea of cellulose, what it's like, and the fact that it's very hard to digest. The important thing is that only monosaccharides can be absorbed. So polysaccharides, disaccharides all have to be broken down into a monosaccharide before they can be absorbed. So let's take a look at how starch is digested. First of all, starch is, again, a polysaccharide. It contains many, many glucose monomers, which each of these little hexagonal shaped structures is a molecule of some sort of carbohydrate, some sort of sugar. The enzymes for digesting starch are salivary amylase and pancreatic amylase. The end products of breaking down these large chains of polysaccharides get broken down into disaccharides, primarily maltose, or what are called limit dextrins, which are smaller chains of sugar molecules. So the disaccharides are just doublets of sugar molecules, and limit dextrins are longer chains of sugar molecules. So these have to be further digested down because, as I said, we can only absorb monomers of what are called simple sugars. So the enzymes of digestion, dextrinase breaks what are called limit dextrins down into glucose. Glucoamylase breaks polysaccharides down into glucose. Sucrase breaks sucrose down into its constituents, which sucrose, remember, is a disaccharide, as is lactose and maltose. So sucrose is broken down into its two component sugars, that is, molecule of fructose and one of glucose. Lactase breaks lactose down into a molecule of galactose and a molecule of glucose. And maltase breaks a molecule of maltose down into two glucose molecules. These enzymes are all what were called brush border enzymes because they reside in the brush border of the small intestine. Once these simple sugars are broken down into, disaccharides are broken down into simple sugars, that is, carbohydrate monomers, they absorbed through transport from the lumen into the bloodstream. And I'll show you, we'll bounce back and forth between this slide and the next. So on the next slide, we actually show, this is the same figure we saw a couple slides ago with our brush border here. And in this case, we're concentrating on the absorption. So we've got the breakdown, in this case, of maltose into 2 sucrose molecules by maltase. Remember, it's a brush border enzyme. But of course, then the glucose is going to have to be transported from the lumen into the bloodstream. So we have this active transport. Glucose and galactose are absorbed by secondary active transport across the apical membrane and facilitated diffusion across the basolateral membrane. Fructose is absorbed by facilitated diffusion across both membranes. So we'll take a look at these processes here on this next slide again. So we have here our good old friend, the sodium-potassium ATPase. And this is, of course, pumping sodium out of the cell and potassium into a cell, which means that we are reducing the sodium concentration within the cytoplasm of the cell because we're pumping it out. This sets up a concentration gradient where there's more sodium out here in the chyme, and it is moving down its concentration gradient because there's more of it outside the cell than there is inside the cell. And this is a cotransporter, that grabs the glucose molecules and pumps it through along with the sodium. So sodium's moving down its concentration gradient, and the pump is grabbing glucose molecules and pumping them through as well. And then, of course, they are being moved by other transport systems from the cytoplasm into the interstitial fluid and ultimately being absorbed into the blood flow. Let's take a look at the digestion and absorption of proteins. Typical diet consists of 125 grams per day, which far exceeds our actual requirement of only 40 to 50 grams per day. Proteins to be digested and absorbed include those that are consumed in the diet. In addition, those that are secreted into the lumen of the intestinal tract. If you think about these various enzymes and other hormones that we'll be talking about later on that are involved in the regulation of the digestive process, these are themselves proteins. So, of course, they can be digested and absorbed ultimately. As well as there's proteins involved or that are contained in the cells within the lining of the intestinal tract that are sloughed off. And these will also be absorbed and, digested and absorbed. The products of this digestion include amino acids, dipeptides, and tripeptides. Just as we saw with carbohydrates, we have very simple breakdown products, in this case, single amino acids, or chains of amino acids. Short chains such as dipeptides, which is 2, and tripeptides, which is, of course, 3. So the enzymes that break down proteins are referred to as proteases. And there are 2 basic types, endopeptidases and exopeptidases. I took this figure from a different physiology book than ours because ours does not have an illustration of this process. So here we have a fairly simple protein. This is a polypeptide chain. It has a head end, the amino terminal, I should say, and the carboxy terminal, which is the tail end. So with this, there is a definite head and tail end of these molecules, unlike what we see with carbohydrates, and this is important because of these different enzymes. Endopeptidases break the chain down between the head and the tail. They work toward the center portion of the polypeptide chain or the protein chain and break this long chain down into shorter chains. But they're working at the center point or near the center of the amino acid chain. Hence, they're called endopeptidases because they're working within the molecule. Exopeptidases target the head or tail end. And that's why they're called exos, because they're working on the outside end of the polypeptide chain. Whereas the endopeptidases produce shorter amino acid chains, the exopeptidases produce, by and large, single amino acid chains that, of course, still have a head and tail end. So you have to have the endopeptidases breaking large chains down into smaller chains, and ultimately the exopeptidases are going to be breaking those down into single amino acids. So in the stomach, protein digestion, first of all, begins in the stomach. There is no protein digestion going on in the mouth. The mouth is only involved in the digestion of carbohydrates. And the enzyme that is responsible is pepsin. It is secreted in an inactive form. Remember we talked before about zymogens. It is secreted as pepsinogen, and pepsinogen is activated by the acid environment of the stomach. And again, we talked about this in the last lecture. So here we have the chief cells of gastric gland that are producing pepsinogen, and the parietal cells, which are producing hydrochloric acid. Now, pepsinogen, again, is the inactive form. It becomes pepsin. But it's a little bit complicated in how this happens. The acid activates the pepsinogen. If you notice here, the pepsinogen has an asterisk next to it. This is an activated form of pepsinogen, which then will auto-convert pepsinogen that was not activated into pepsin. So pepsinogen becomes activated and then converts other pepsinogen molecules into pepsin. And this, of course, requires this acid environment to do this. And then, of course, the pepsin helps to break these protein chains down into smaller peptide fragments. Within the small intestine, we have pancreatic proteases. There are three. There are trypsin, chymotrypsin, and carboxypeptidase. Trypsin converts chymotrypsinogen into chymotrypsin. So, of course, trypsin is important for the production of chymotrypsin. It also converts procarboxypeptidase into carboxypeptidase. So, of course, the trypsin is important then for the production of both of these digestive enzymes. And that is shown here. We have the enterokinase, which is an enzyme that breaks down the precursor for trypsin, which itself has a precursor, trypsinogen, breaking it down into trypsin, and then, of course, activating these other two enzymes. Again, the concept of the zymogens. We also have brush border proteases. We have the brush border enzymes in the stomach. We have brush border proteases in the small intestine. And these include the enterokinase, which converts the trypsinogen into trypsin. That's what was shown here. And aminopeptidase. Excuse me. So I mentioned the concept of zymogens. Again, just as a refresher, it's an inactive storage form of proteases. These are three examples. These are stored in zymogen granules, that is, vesicle-like structures within cells that are secreted by exocytosis and then themselves are activated by proteolytic activation. So we have enzymes that are activating other enzymes. So there's a review again on pepsin. Chief cells secrete pepsinogen, which is the inactive form. The hydrochloric acid cleaves the pepsinogen into pepsin. And pepsin, of course, is the active form. And the parietal cells secrete the hydrochloric acid. So it is the digestive activity itself that is helping to digest the food, that is also helping to activate the enzymes of digestion. So absorption of amino acids. Again, this diagram was taken from another physiology book because your book does not have an illustration showing this. But this is fairly similar to what we saw with carbohydrates in the stomach. It's just in this case we're talking about amino acids. First of all, they have to cross the apical membrane by a sodium-linked secondary active transport or by facilitated diffusion. So this is what's going on at this level of the cell, and cross the basolateral membrane by facilitated diffusion. So that's what's going on here. So we have our long polypeptide chains that are protein broken down into shorter chains. And as we show here, there is a proton exchanger that can take up very simple dipeptides and tripeptides. These may, in fact, continue to be shuttled through the cell and exchanged again with another proton exchanger here and brought into the circulatory system. Far more of these, though, will be broken down by peptidases that will convert these to single amino acids that is then going to go through with a sodium cotransport, entering the same stream that is coming in of single amino acids that are being transported here as well through the same process. And then peptide fragments, that is, chains of several amino acids that have not been broken down yet, can be brought into the cell by endocytosis and put into a vesicle, and then through the process of transcytosis, that is, transportation across the cell, these small peptide fragments can also enter the circulatory system. So dipeptides and tripeptides, basically the double or triple amino acid chains crossing the apical membrane by active transport, that's what we showed here, broken down inside the cell, that is, these are the cells lining the small intestine, into single amino acids, and then those cross the cell membrane into the circulation by facilitated diffusion. Now, lipids are by far the most complex molecule from a standpoint of digestion and absorption. First of all, the typical diet contains about 25 to 160 grams of lipids. These are what we refer to as fats in the diet. 90% of these lipids are triglycerides, and triglycerides are a certain structural variant of lipids. Lipids face a special problem in the digestion and absorption because they're not water soluble. So they do not mix with the contents of the stomach or the intestine very well because of those contents, of course, are aqueous, meaning that they are lipophobic. Therefore, lipids tend to form fat droplets, and of course, because they don't mix with this aqueous environment, it's very difficult to digest them. So what we have to do is to find ways of breaking these droplets down into smaller droplets and ultimately in overcoming their hydrophobicity. So there are enzymes that will break down lipids into their individual molecules, but in order to do that, we have to overcome the hydrophobic nature of lipids. These lipases are secreted by the pancreas, but they can only act on molecules near the edge of the fat droplet. So what we do, or what the body does, is it secretes bile salts. We referred to these in a previous lecture. And these increase the surface area of the droplets by breaking the large droplets down into several small droplets. And this is the process of emulsification. So we have, with bile salts, they form a hydrophilic surface on these fat droplets, lipid droplets. And by having a hydrophilic surface that is provided by the bile salts, it allows the enzymes, these lipases, to get a toehold, if you will, into the surface of these lipid droplets and help to break them down. So let's take a little bit more of a look at bile salts. So an illustration of their structure. They themselves are what we call an amphipathic molecule. That is, they have a hydrophobic and a hydrophilic side. And, of course, the hydrophilic side will interact with the aqueous environment, that is, the enzymes, lipases, and such. And the hydrophobic side is pretty much acts just like the lipid itself. Bile salts are synthesized in the liver from cholesterol. And that means that they themselves have a lipid component or a lipid moiety. They are secreted into the bile, into the duodenum, where they start the emulsification of fat. And again, emulsification is the breakdown of large droplets into smaller droplets. So let's take a look at further steps on this emulsification process. We have a fairly large droplet of lipid here, and of course it does not mix with water. So these bile salts, remember they have both, we'll go back here, the hydrophilic side and a hydrophobic side. So the hydrophobic side will, of course, associate more readily with the lipids themselves, and the hydrophilic side will be oriented outwards towards water. So we have much more surface area exposed to the lipases, which, of course, themselves are hydrophilic. So then what happens is that these smaller droplets get broken down into what we call a mycele. And this is the process of breaking triglycerides down into a monoglyceride. So, of course, you have 3 being broken down into 1, and 2 fatty acids. So some of the fatty acids and monoglycerides are absorbed, but others form micelles, and that's what we're showing here. So the mycel is going to be a short-term storage form of lipids, whereas the monoglycerides and fatty acids themselves are getting absorbed. These are other lipids that are being held in storage, very much like the idea of glycogen for storage of carbohydrates. And there is an equilibrium between the micelles and the fatty acids and monoglycerides. We show that in this next diagram. Actually, sorry, a couple more diagrams. Let me jump ahead. So here we have the monoglycerides and fatty acids, which can be absorbed. But there's also micelles that are being formed from these individual molecules. And these act as a reserve for the monoglycerides and fatty acids. So as the amount of digested lipid material declines, we have fewer of these molecules that can be absorbed. The micelles will themselves break down, forming these monoglycerides, fatty acids, which will, of course, help to sustain prolonged absorption in the absence of a continued supply of the raw fats coming in. So there is an equilibrium between these free fatty acids and the monoglycerides and those that are stored in the micelles. The free form can be absorbed by simple diffusion. That's what I was showing here. These can be absorbed by simple diffusion. Remember, because they are themselves lipid in structure, just very small lipid, they, of course, can cross the lipid membrane. Inside the epithelial cell, they're going to enter the smooth endoplasmic reticulum and reform into triglycerides and other lipids. Those lipids then will enter the Golgi apparatus, be packaged into chylomicrons. So let's jump ahead in this picture here. Actually, it's a couple more pictures. So we're going to have the monoglycerides, fatty acids coming in and being processed by the smooth endoplasmic reticulum back into triglycerides, which are then going to be processed by the Golgi apparatus. And we are going to have these vesicles coming out of the Golgi apparatus that we call the chylomicrons. These will be secreted by the cells by the process of exocytosis into the interstitial fluid, and then they will enter the lymphatic system via the lacteal. We'll talk about that in a moment. Let's go back to our picture. So we have our chylomicrons, which again are basically small lipid droplets, just highly processed. So these, again, because they're hydrophobic, are not going to be able to get into the bloodstream. These will be absorbed into the lacteal. Remember, the lacteal is part of the lymphatic system. And so they will be absorbed that way, whereas the other simpler fats will be directly absorbed into the bloodstream. So I've shown this diagram already in terms of how lipases work. And then again, this one in terms of absorption of fats. Now let's move ahead to the reprocessing of bile salts. We've talked about how important they are in terms of emulsification of fats. But rather than synthesize new bile salts, there's actually what's called an enterohepatic circulation. And do not confuse this with as the same thing as the hepatic portal system. This is in addition to it, but still using the same circulatory bed. So we have the bile salts that have been released by the liver through the common bile duct, helping to break down fats. After the fats are broken down, the bile salts no longer have lipid droplets to bind to, so they are absorbed in the mesenteric circulation and transported through the hepatic portal vein, along with everything else that's being absorbed by the hepatic portal circulation. So as I said, it's in addition to everything we've said about the hepatic portal system before. So we've talked about absorption of carbohydrates and proteins and lipids. But of course, there's other things in our diet. There are vitamins. Some of the vitamins are fat-soluble, meaning, of course, they're hydrophobic but lipophilic. These include vitamins A, D, E, and K. These are absorbed along with lipids. So they dissolve in lipid droplets, micelles, and chylomicrons, and that is how they are absorbed. Whereas the water-soluble vitamins require special transport proteins to bring them from the lumen of the GI tract into the bloodstream. Vitamin B12, I've mentioned in the last lecture, it is absorbed only when bound to intrinsic factor. An intrinsic factor is a substance that's located within the lining of the gastrointestinal tract that is required to bind vitamin B12 to allow it to be pumped into the circulation. So let's take a look at the absorption of other minerals. In this case, we're talking about sodium, chloride, and potassium. Sodium is absorbed with what's called solvent drag with water reabsorption. And let's take a look at that here. We have, it's not actually shown, but you have water molecules that are, of course, being absorbed from the lumen, and sodium is going to be dragged along with that. And it's just a matter of, just as we talk about in osmosis, water moving along with solute, that we can also have solute moving along with water in this case. As well as sodium can be actively absorbed in the jejunum, ileum, and colon. Chloride is absorbed passively, and it's following sodium reabsorption. So we have sodium and chloride coming together. There's also potassium, which is passively reabsorbed. I should say passively absorbed, and it can be secreted when the luminal concentrations are very low. So if there's not enough potassium here in the chyme, we're actually going to have some potassium that is going to be secreted as well. So potassium can go both ways across the lining of the GI tract. Calcium is a little bit of a special case. It is actively absorbed in both the duodenum and the jejunum. And there is a protein called calcium binding protein, which is in the brush border of these parts of the small intestine. And the calcium is transported into the epithelial cell by this calcium binding protein. And then it is transported out of the cell across the basolateral membrane by a calcium pump. Vitamin D3 when it's fully activated, has this chemical structure, which is referred to as 1,25-dihydroxycholecalciferol. Just easier to think about it as vitamin D3. This increases calcium absorption by increasing the concentration of this calcium-binding protein. So it's why vitamin D3 is so important for calcium absorption. Iron, another mineral, is absorbed, again, by a fairly complex process. A protein called transferrin is secreted by enterocytes into the lumen of the small intestine. And it binds iron, forming a complex that then binds to a receptor. This receptor then allows this transferrin iron complex to be taken into a cell by receptor-mediated endocytosis. Some of the iron will be stored in the enterocyte itself as ferritin, and some will be transported in the blood. And it's transported in the blood bound to transferrin. So transferrin plays a dual role, getting the iron into the enterocytes, that is the cells lining the GI tract, as well as transporting iron within the blood. And we talked about bicarbonate being secreted as a way of helping to protect the lining of the stomach from the acid environment, as well as in the small intestine to help neutralize the acidity. And I mentioned in passing in a previous material that the bicarbonate is actually reabsorbed as a way of recirculating it. So in the jejunum, the bicarbonate is absorbed passively. And in the ileum and the colon, it is secreted in exchange for chloride ions. So again, it is both absorbed and secreted within the lower portion of the GI tract. And of course, all of our water that we consume was going to have to be absorbed through the GI tract ultimately. About 2 liters a day taken in, but a dramatic amount is actually secreted into the GI tract, 7 liters of secretion. So while we only actually consume about 2 liters of water a day, we secrete 7 liters of water as various enzymes and other digestive secretions into the GI tract. So of course we're going to have to reclaim a fair amount of that, otherwise it becomes severely dehydrated. Water absorption is passive. It follows the absorption of solutes by the process of osmosis. And much of this is of course occurring in the small intestine, but what is left in the material that's entering the large intestine will then be absorbed from the large intestine.